Hi Alex, tell us a little about yourself and what is your background and what drew you into AI research?
Yes, sure. Thanks very much for having me. I'm Alex Gavryushkin. My background is in computer science and mathematics. I've written a PhD on limitations of algorithms, including AI algorithms, back in 2009. And since 2013, I have been working on applications of AI in biotechnology, including drug discovery. My last academic appointment was as Associate Dean of Research at the University of Canterbury. And 2 years ago, I started a drug discovery company, RNA targeting drug discovery company in Sydney, which I'm the founder and chief executive of. I'm also an honorary professor at the University of Sydney's AI and Drug Discovery Institute and a visiting faculty here at Victoria University.
Thank you. AI is everywhere these days, but what is your particular area of expertise?
My particular area of expertise is AI for biological applications. These include evolutionary and systems biology generally and drug discovery in particular. So we're building algorithms that, given an RNA target, predict either ASOs or small molecules that are great candidates to become drugs eventually. So the way we develop our systems is to optimize for a molecule to become a great drug rather than focusing on any specific types of optimization such as structure or binding or anything like that.
Yeah. So many people know AI through tools like ChatGPT. How is the type of AI you work on different?
So when people develop AI models, it's very important to choose so-called optimization functions. So to train your AI to get good at a particular task. And with your example of GPT-like models, the way they're trained is by learning how to very accurately predict the next token. So in the example of text generation, that would be the next word in a sentence. In genomics, that would be the next nucleotide, for instance, in a sequence, or the next amino acid in proteins. So that's the objective function, right? So, and your model can get really good at generating human-like text or sequences that look like genomic sequences. And then you can do so-called fine-tuning of those models. For instance, in the case of ChatGPT, you can fine-tune it on question answering, and then the pre-trained model becomes a chatbot. In a biological application, AlphaFold 3-like models will be trained on predicting structure, and that is their objective. So when you do drug discovery, predicting 3D structure of the protein is an important task, but it doesn't necessarily lead to discovering great drugs, because after all, the working molecule would be the ligand that you're designing for your protein, or the molecules we are designing to bind our RNA. So in the context of drug discovery, you don't necessarily need to solve these very complex problems, such as the RNA structural dynamics, to predict great drugs. So the fundamental difference between different type of approaches, including the one that we are developing, is what you're optimizing for. So what we are working on is so-called multi-objective optimization. So we select several readouts that are relevant to drug discovery. And they include toxicity, they of course include binding, but they also include later stages of the drug discovery process, such that the function of the molecule in mouse models, in larger animals, and eventually in clinical trials. If you are working on this topic commercially, then freedom to operate is an important objective. And that also can be included into the multi-objective optimization. So you can be designing molecules that you can patent and progress towards the clinic. So it's important to understand for drug discovery process that some of these other objectives can potentially be a lot more important than solving basic scientific problems such as protein structure prediction. For instance, manufacturability is such example where if you are in a real scenario and you have access to real labs or CRO partners, then you will want your predictions to be synthesizable and manufacturable at the right scale and purity by those partners. These are again something that you can include in AI optimization and that can potentially make the optimization more efficient without solving the very hard problems such as the RNA structure dynamics.
Why is AI becoming such an important tool in RNA research?
Because it's hard. Because traditionally, when human intellect has not been supplemented by the powerful computational tools such as AI that we have today, we were bound to predicting and generating the molecular assets and drugs that humans can comprehend with our intellect. However, the biology is so complex, and the function of RNA is only recently has only recently become something people started working on. Traditionally, the role of RNA has been just a messenger that sends information from the DNA to the protein. So people have been focusing on DNA and protein. Now it's common knowledge that there is a lot more that RNA does in the cell. And the complexity of those processes can be higher than what human brain can comprehend. And this is where AI systems come into play, because they can be doing the multi-objective optimization that I mentioned. They can be trained to learn from heterogeneous data types. It's called multimodal type learning, where we don't really know what type of data is the most relevant for the drug discovery process. But by training large enough models on diverse enough data, we can enable those AI systems to pick up the right signal and the most informative data modalities.
The next question is, where do you think AI can make the biggest impact in developing new RNA vaccines and therapeutics?
I think the biggest impact of AI in drug discovery in general and RNA therapeutics specifically is the number of experiments that need to be performed at cost and time in the lab. So by training AI on existing data and by letting AI to predict the missing training data, we can significantly reduce the number of molecules that need to be synthesized in the lab, the number of assays that need to be run in the lab, and the number of animal experiments that need to be performed. I think that will be the biggest impact. And potentially eliminating animals from this process altogether is something that is within reach.
Yeah. What are you hoping to achieve through your work with RNA platform?
My work with the RNA platform is a partnership between the platform and my company, Fold AI, where we are developing algorithms and train them on our proprietary data as well as the data that is being generated by the platform with the objectives that are relevant to the platform. And the KPI and the goal for us to achieve here is the goal that I just mentioned, reduce the number of experiments. So we are collaborating with different pillars across platform, identifying areas where AI can make a significant enough contribution that it's worthwhile investing into the development. For instance, if we can reduce the number of experiments required to optimize an mRNA molecule by 10x, then whatever is saved is worthwhile investing in developing of AI. And mRNA optimization is one example, but there are numerous examples anywhere from target identification to clinical trial design. So we're exploring this in the context of New Zealand, in the context of the RNA platform, where is that value proposition is sitting for AI to be developed, for specific AI to be developed, and where existing tools can be deployed. And we also are supporting the teams with help around commercially available, powerful enough algorithms that are relevant for the work happening at the platform.
If I'm an RNA researcher, when should I come and talk to you?
Well, the best time to talk to computer scientists and AI expert for an RNA researcher is before they becoming one. And the second best is now. And the reason for that is biology is becoming more and more digital, and the role of computational tools and approaches is extremely hard to overestimate now. It was actually quite interesting during my time in the academia. I have been working on potentially setting up a computer science course for biology students. And that didn't go ahead for the reason that there wasn't enough biology students interested in taking such a course. It was a long time ago. The short survey that we did identified the main issue as being I hate mathematics and computer science, and this is why I'm studying biology. So that basically demonstrates the fundamental problem that many biology-trained experts face now, when it's impossible to discover new knowledge in biology without reasonably solid understanding of what algorithms can enable. It's impossible to design new experiments effectively without doing computational simulations. And the applications of computing in biology go far beyond that. So to answer this question in an actionable way, I would say that the best time to collaborate is at the experiment design stage. Basically, for the experiments where there is human knowledge and expertise that is 100% certain in terms of what experiments need to be done to answer that particular question, the role of AI is minimal. However, the vast majority of exploratory work, including in drug discovery, requires a healthy level of guessing what experiments to do, what modifications to test, and so on. And that's where AI becomes extremely powerful, because by training AI on already existing models with the objective that is relevant as the outcome of those experiments, we can significantly reduce the training data that will eventually be required to answer that question. We will be able to answer many of those questions computationally and only use wet lab work for validation. So experimental design and training data generation design are the most valuable steps in biotechnology and drug discovery where AI people can be involved. It's often not the case, and it's often that the data has already been generated, in which case managing the data and maintaining all the records of all the measurements will be also extremely important and still a way to work and collaborate with us. However, depending on how well the data science practices have been followed during the data generation process, it might require more or less data science work before the data is ready for AI training and processing. And because the vast majority of data available out there has been generated without AI or computer science in mind, AI-first startups like ourselves spend absolutely significant amounts of time on doing this data processing and data preparation. So to maximize the impact of AI today, the best way to engage is at the time when the experiment is being designed.
What kind of research questions are best suited for AI?
Yeah, that's a really good question. And I don't think there is an easy answer to that. I don't think we can classify research questions as suitable or non-suitable for AI. Obviously, with the way things are developing, especially in biology and in systems as complex as human organism, the most appropriate and interesting questions can be those where humans don't have a chance to understand the process, where we reasonably believe that the prediction task or generation task is complex enough that it will never be comprehended by humans, and just trust that to AI. And I know there will be some healthy pushback to a view stated like this. But if we go back in the past and think of the very first computational machines like calculators that had also had reasonable skepticism before they became adopted, then we realized that today is impossible to question basic computing performed by machines. It's going to be like that with biology, where we will just give up on some of the biological systems that are too complex to understand, and we will have reliable and trustworthy AI approaches that model those systems, simulate those systems, and answer questions.
What information do researchers need to bring to start working with AI team?
Well, the key initial aspects of any collaboration between scientists and AI engineers is training data and the question that they are trying to answer using experiments or computational approaches. As I mentioned, it's fundamentally important. It's of fundamental importance to identify the correct optimization function, to identify what is the parameter or what are the parameters that you want to maximize or minimize. Once that is identified, AI approaches will be in business. The next step will be whether the training data is available out there, whether the models that have been trained on the existing public data, for instance, have a chance of generalizing to the questions that are being answered. And usually the cutting edge research, and in fact the difference between cutting edge research of today and not so cutting edge, is whether the data that is being generated, the questions that are being answered, are out of distribution enough for the existing AI models, because we still don't have AI models that generalize on all of biology, but we have extremely powerful ones that do generalize to a lot of things. So generating the data that is within the distribution that has been learned by those models is not very interesting. And that's an important engagement with the AI researchers to understand whether the existing tools cover already this sort of questions. However, for the cutting edge research, the questions will be far away from the existing tools and AI to generalize towards. So then the question becomes, what is the data that is available out there that would allow us to bias existing tools towards that particular data set, or whether there is capability of generating enough training data to extend the distribution that was learned by the AI towards the questions and data points of interest for the particular researcher. So yeah, to summarize, it's the objective function, the measurements that they expect from the experiments and the training data.
So why should researchers work with your team instead of just using off-the-shelf AI tools?
The questions that can be answered using existing tools are not cutting-edge research, for the reason that the models that are available to anyone in the world, they don't generalize across biology. They've learned particular distributions and they can answer particular questions that can be considered as settled questions. They don't require generating novel data. Just to give an example, predicting protein folding. We have a reliable and good enough models that go from sequence to the 3D structure. It doesn't really make sense to go in the lab and keep on validating these models. However, for some of the aspects of that work, the questions can be out of distribution. They can be something that AI does not generalize on. And this is where no tools are available. It doesn't mean that AI cannot answer that. It means that novel AI tools need to be developed to work for the specific application. And it's something as directed towards the outcome as drug discovery. This is of the biggest importance, simply because tools that are available off the shelf oftentimes are solving a fundamental scientific problem that is extremely important to solve, but they are not directed towards a specific application. And as an example of that, I can suggest, for instance, the quality control in mRNA vaccines. So when an mRNA is synthesized, there is a lot of parameters that people measure and control the quality of the molecule of. However, the models that have been used to generate that sequence were never aware of those parameters with respect to which their quality will be assessed. So they don't have a chance of learning that. The things that the models have learned is important for drug discovery, but there are other things that researchers will be looking at. So there is no way around this but designing a custom model if we don't want to perform everything experimentally and basically sampling these extremely big space uniformly at random.
That totally makes sense. And we're moving on to the question about multimodal AI. And the question is, you've mentioned multimodal AI. Can you explain what that means in simple terms?
Yes, absolutely. So one of the first striking applications of multimodal AI that was released and when it became mainstream was basically connecting text under images to images. So if you think of a scientific paper, there will be a caption, there will be a figure and a caption. And traditionally, AI models that have been designed for text and images have been different. Different AI architectures, different training approaches, so it's completely independent algorithms. But what is obvious is that there is information that is relevant to the image in the text around it, and vice versa. There will be information in the image that is relevant to the text. So people came up with a way of training simultaneously on both images and text. And this is what multimodality is in a nutshell. When we have different data types that we use for training a single unified model. It can be images and text, it can be video, audio streams like in a movie and so on. The popular example of multimodality working at extreme is self-driving cars that use many different sensors. So there are a lot of approaches to self-driving, so some are based just of images, but there are those where data streams will be coming from different sensors, and those data streams would be of different importance to the outcome and different relevance, and the models that will be learning from all of these data types will be the most powerful, because you don't need to prescribe of how to combine those things. In biotechnology and drug discovery, there is usually no obvious way of how to combine these different types of data. And usually that will be different types of experimental data. Sequences, structures, binding experiments, those assays that I mentioned, they all will be generating different data types. And the multimodal approach allows training AI systems that will be learning from these different type of experiments, different data types simultaneously rather than independently. Traditional approach would be to train an AI model for each, make the predictions, and use some classic algorithm type to combine that. For some applications that will make sense. For others, where we don't really have a good understanding or will never be able to understand where the main signal is coming from, it's best to allow the AI to make this decision based on training.
What does a collaboration between your team and an RNA researcher actually look like?
In my experience, in an academic setting, the most efficient collaboration between experts as diverse as a computer scientist and an RNA biologist, executed in the most efficient way, would be through team members working alongside each other on a predefined project. When a computer science PhD student is developing network architectures, algorithms, training methods, while the RNA biology expert supports that with providing access to the training data that is publicly available, helping navigate what can be obtained from public sources, as well as what type of data can be generated. So when this collaboration is happening from the early stages of the project, when experiments are being designed by the RNA scientist group and algorithms are being designed by the computer science group, academically, in my experience, is the most efficient way to produce the results, but also to produce these unicorn-type experts who are literate in both fields. Because during this process, the computer science team members, can be PhD students or postdocs, will be learning the language of RNA, will be learning things about biology that are very well understood and 100% correct, that AI doesn't need to learn and can take as given. While the RNA scientists will be learning about AI capabilities, what can be achieved using off-the-shelf trained models, what can be achieved by taking existing architectures and training them on the data that they have, and when new architectures are required, because this is such a novel and dynamic field that for cutting-edge research, it's not far from getting to the stage where both fields need to innovate to achieve the result. And this, in my experience, is the most fruitful and satisfying type of work, when innovation needs to happen in both AI, computer science, and RNA to achieve the common goal. From the business perspective, however, the most fruitful collaboration would be to identify the training data, to identify the questions, and to execute it in the most efficient way.
Looking 5 years ahead, what would success look like for our AI within the RNA platform?
The number of wet lab experiments reduced by 10x is what we can reasonably expect from deploying powerful AI systems throughout the platform and throughout the process of drug design. Reducing the number of experiments and screens and assays that needs to be done experimentally is the fundamental value proposition that is absolutely within the reach and that is absolutely happening both commercially and academically. However, the ultimate goal of our collaboration and the platform in general is drugs on the market. And the deployment of AI that we are envisioning as part of this collaboration will shrink down the timeline between discovery and deployment. We shrink down timeline required for successfully responding to a viral outbreak. Will shrink down timeline required for other types of vaccines and drug discovery. Will enable personalized approaches when the timeline between diagnosis and drug being designed, optimized, manufactured, and delivered becomes real.
What do you mean by 10x?
I mean, out of 10 experiments that are being performed randomly by guesstimating what modification to the RNA to make and what to test, there will be just one that is actually required. Mm-hmm. So eliminating 9 out of 10 experiments that are happening at random.
Mm-hmm. That's what AI can help.
Yes.
Hopefully. You don't have to answer this question, but it says, if you could solve one big challenge in RNA research using AI, what would it be?
It will be an AI-predicted drug that makes it through the clinical trials and delivers outcome for the patients that don't have any cure now.
That's amazing. And we have 2 closing questions. That will be the end. 17 is, what excites you most about being part of RNA platform team?
I think it's the combined effort of all the expertise across the country directed at something as important as drug discovery in the context of New Zealand, relevant to New Zealand, relevant to the patients.
Finally, what message would you like to share with researchers who are curious about using AI but unsure where to start?
I think people should not be approaching AI for the sake of using AI. So I don't think we need AI just for the sake of it. I think identifying the problems that we are trying to solve is far more important, and going off the ultimate goal we are trying to achieve. What is the aim here and how we can get there? And if we have this pathway and towards the goal, and we we have a plan of what needs to be done to achieve that, then the question will be, what are the steps on this pathway that can realistically be eliminated by automation, by computing, and by AI? And with that question, it's a lot easier to answer simply because some of these tasks will be easily automatable and have not been automated up until now simply because there was no tools. And now with the pace of AI progress, the things that can be automated expanding extremely quickly. But more interestingly, it's the questions where human expertise suggests that the answer is somewhere in the data. We just don't know how to extract. And that's, I think, is the most exciting entrance to AI. And this is where AI researchers will be required, simply because it's not a task that is obviously automatable. But to start using AI, I would suggest first to eliminate the steps that are obviously automatable, even if it requires a little bit of friction initially. Because the human nature is to opt out for something that we know how to do, and we can do as fast as we can. With automation, it's not always the case that you get the benefit of reaching your goals more efficiently when you first try it. But if it's something that will be required again and again, that's where automation can help. And AI systems, AI agents are becoming better and better at automating many different tasks throughout the scientific process. So identifying that is the first step.
You mentioned about data, that negative data is quite useful, and you are interested in working with those data. Could you explain more about that?
Yes, absolutely. When I was talking about availability of training data for a particular task, RNA sequence optimization or whatever that can be, there will be plenty of data available out there that has been published, made commercially available, or exists otherwise. Again, human nature is to talk a lot about successes and not much about failures. And the academic system that generates the vast majority of the training data for drug discovery and for science that is publicly available is designed to reward positive results. If you find a molecule that reliably binds to the target, you can very easily publish that and get the rewards you expect. The fact that to get there you've tested thousands or maybe millions in many cases other molecules often is left aside. But that type of data is of absolutely massive importance for AI to develop the human level expertise. And this is the type of data that effectively enables human expertise. If you're a professor working on a handful of targets all your life, you've generated so much negative data that you have the pre-trained model somewhere in your brain that basically suggests that these are the promising things to try. The AI training is not dissimilar to that. Without knowing what has been tried and has failed, AI will struggle to generalize to that part of of the search space, that part of the distribution. So to develop systems as powerful as possible, no data should be lost in this process, and the more negative data that is made available to AI researchers, the better.
Thanks for your time, and hope to see you soon, and definitely at the RNA Symposium.
Yeah, thank you very much for having me. Yeah, looking forward to continuing this collaboration and to the RNA Symposium as well.
Yeah. Thank you.
Thank you.